Welcome to AP Daily. This is Mr. Burris from Union High School in Vancouver, Washington. Today's topic is 3.3, Environmental Impacts on Enzyme Function. So what will we learn? In today's lesson, we're going to look at what does it mean for an enzyme to be denatured? How do changes in pH or temperature affect enzyme activity? How do changes in enzyme concentration affect the rate of reactions? How do changes in substrate and product concentrations affect the rate of reactions? And finally, how do competitive and non-competitive inhibitors affect enzyme activity? So let's dive in. Enzymes have a unique functional 3D shape known as the conformational shape or tertiary structure. Changes in the conformational shape of the enzyme equals denaturation. Changes in the environmental temperature can lead to denaturation. Changes in environmental pH can lead to denaturation as well. Enzyme denaturation is typically irreversible, and the catalytic ability of the enzyme is lost or severely decreased. However, in some cases, enzyme denaturation is reversible, allowing the enzyme to regain catalytic activity. So what does it mean by optimum temperatures? Well, optimum temperature is a range in which enzyme-mediated reactions occur the fastest. Reaction rates change when optimum temperatures aren't maintained. When there's an environmental increase in temperature, there's an initial increase in reaction rate. This causes an increased speed of molecular movement and an increased frequency of enzyme-substrate collisions. Temperatures increases outside of the optimum range result in enzyme denaturation. When the environmental temperature decreases, it generally slows down the reaction rate, as shown on the chart. It leads to decreased frequency of enzyme substrate collisions. However, it does not disrupt enzyme structure, so no denaturation occurs as the temperature decreases. So what about pH? pH measures the concentration of hydrogen ions in a solution. It is measured on a logarithmic scale. A logarithmic scale, or log scale, is a way of displaying numerical data over a very wide range of values in a compact way. So, small changes in pH values equate to large shifts in hydrogen ion concentrations. So, for example, a pH of 6 has 10 times more hydrogen ions in solution compared to pH 7. So what about optimum pH? Well, it is a range in which enzyme-mediated reactions occur the fastest. Changing the pH outside of this range will either slow or stop enzyme activity. Enzyme denaturation can occur as a result of increases and decreases outside of the optimum range. Changes in hydrogen ion concentrations can disrupt hydrogen bond interactions that help maintain enzyme structure. So how do the concentrations of substrate and products affect reaction rate? Well, an initial increase in substrate concentration increases reaction rate. More substrates mean more opportunities to collide with enzymes. Substrate saturation will eventually occur, as you can see on the chart. This results in no further increase in rate. The reaction rate will remain constant if saturation levels are maintained. However, an increased concentration of products decrease the opportunity for addition of substrate. Matter takes up space, so the more product in the area means lower chance of an enzyme substrate collision, and that will slow the reaction rate as shown on the chart. So how does enzyme concentration impact reaction rate? Well, first, changes in enzyme concentration can also impact reaction rate. Less enzyme equals slower reaction rate. So how does enzyme concentration impact reaction rate? Well, first, changes in enzyme concentration can also impact reaction rate. Less enzyme equals slower reaction rate. There is a less opportunity for substrates to collide with active sites. However, More enzyme equals faster reaction rate. There is more opportunities for substrates to collide with active sites. So competitive inhibitors, they are molecules that can bind reversibly or irreversibly to the active site of the enzyme. They compete with the normal substrate for the enzyme's active site. If an inhibitor concentrations exceed the substrate concentration, the reactions will slow down. If inhibitor concentrations are considerably lower than the substrate concentration, reactions can proceed normally. If an inhibitor binding is irreversible, the enzyme function will be preventable. However, if an inhibitor binds reversibly, the enzyme can regain function once the inhibitor detaches from the active site. Enzymes can have regions other than the active site to which molecules can bind. These are called allosteric sites. Non-competitive inhibitors do not bind to the active site. They bind to the allosteric site. Binding causes conformational shape change to the active site, and the binding prevents the enzyme function because the active site is no longer available. The reaction rate decreases, and increasing the substrate cannot prevent effects of non-competitive inhibiting binding. So now it's time to practice. The skill focus today is argumentation. We're going to predict the cause or effect of a change in or disruption to one or more components in a biological system based on data. So let's take a look at the question. So for this question, I would like you to push pause and then first look at the data table and ask yourself, what is it telling you? Next, read the prompt and look for the bold-faced words, which are your task words. And then ask yourself, what is the question asking you to do? Finally, reread the prompt and then answer the question. When you have your question answered, push play again. Welcome back. For today's question, you were looking at making a prediction of the average time it would take for catalase to make a displaced in 1% hydrogen peroxide solution float if the catalase was boiled for 10 minutes before adding to the solution. Your answer should involve that the disc would never float, or it would take more than 30 seconds. So what should you take away from today's lesson? Well, first, denaturation of an enzyme occurs when the conformation protein structure is disruptive, eliminating the ability to catalyze reactions. Changes in pH outside of that optimum pH range in either direction will result in decreased enzyme activity and eventually lead to denaturation of the enzyme. Increasing temperatures outside of the optimum range will initially cause an increase in reaction rates, with continued increasing resulting in denaturing of the enzyme. Decreased temperatures outside of the optimum range, however, will result in a slowed reaction rate, but not denaturing of the enzyme. Increased enzyme concentration, increased reaction rate. Decreased enzyme concentration leads to decreased reaction rates. Increasing the substrate concentration will initially increase the reaction rate, but substrate saturation will not result in continued increase in reaction rate. And then the reaction rate will remain constant if the substrate saturated levels are maintained. For competitive inhibitors, they can bind reversibly and irreversibly to active sites, potentially altering reaction rates. Finally, non-competitive inhibitors can bind to allosteric sites, decreasing the catalytic capability of enzymes. Well, I hope this lesson was helpful. Until next time, thank you.